In this lecture, we're going to talk about molecular biology structures, by which I mean structures in the DNA and other molecules inside the cell that you need to know about to understand the rest of the course. So these are mostly terms and descriptions of functions that go on inside every cell in your body that kind of govern how molecular biology works and how genetics works. So let's start with DNA. So DNA itself, as we've said before, is this very, very long molecule. It's inside every cell in your body. In humans, we have 23 chromosome pairs. So each of those are very, very long molecules of DNA. So the DNA itself, if you stretch it out, would be about 2 meters long inside each cell. Cells are of course, microscopic. So in order to fit inside a cell, it has to be wrapped up in a very, very efficient way. So it's actually wrapped around other molecules called histones, and what you can see is sort of this beads on a string this structure on the left. And those histones are wrapped together in these organized, slightly longer structures. And those together are then coiled up and supercoiled in even bigger structures, which eventually form the chromosomes that you see on the right. So DNA is coiled around itself. The coils are then coiled around themselves in a very complicated way. Now, for DNA to be transcribed and translated, it has to unwrap itself a bit. So we'll talk a little bit more about that next. So another kind of structure, a very different kind of structure, is not so much a physical one, but a sequence structure. And that's what we call repeats. Or you'll hear a lot about repeats in data analysis of DNA. So we just need to know what these terms are. And there are many different kinds of repeats. Broadly speaking, we could classify them in two different types. 10 repeats, which are repeats of identical sequence that occur one after the other. And here's an example of a 5-base repeat, ATTCG, that occurs 3 times in a row. And these, by the way, 10 repeats don't have to be identical, and they can be very long. There are cases where you have sequences that are hundreds of base pairs long that occur thousands of times in a row. And in fact, the centromeres inside of each chromosome comprise largely of repeats that are on the order of 180 base pairs long that occur hundreds of thousands of times in a row. So repeats can get very long and complicated. And we also have interspersed repeats, which really just means the same idea, repetitive sequence that's identical or near identical, but it's scattered around across the chromosomes in widely separate places and across different chromosomes. And these repeats will sometimes cause problems for other types of analysis because they all look so similar to each other. And if you have a very short sequence, remember when we read DNA sequences, we're only reading a few hundred base pairs at a time, so it's not unusual to get a sequence which consists entirely of repeats, and that means we don't really know where it came from because repeats occur all over the genome. So there's also structures of RNA that are important to understand. So a typical human messenger RNA, which is what we get our proteins from, looks like this. We have some sequence, which is shown in green, which is the coding sequence, and sometimes when we talk about mRNA or RNA, we're only talking about what encodes proteins. That's just the green part. But it's important to understand that actually the part that gets copied from the DNA, the transcribed portion of the DNA, which goes into messenger RNA, is longer than that. So typically you'll have some stretch at the beginning of the messenger RNA that's not translated. Then we call that the UTR, or untranslated region. And because it's at the beginning, and remember the beginning is the 5' end, we call that the 5' UTR. And on the other end, we have the 3' UTR, which is usually a longer stretch for various reasons. That's also not translated. And somewhere in the middle is the coding sequence. So we want to know what the protein sequence is. If we can identify the UTRs and get rid of those, then we can read off the coding sequences. From that, we can directly translate our amino acid sequences. And another very important feature of eukaryotic cells, not just human cells, is that they get a poly A tail added to them. So after transcription occurs, after you copy the DNA into RNA and remove the introns, a long series of A's gets added, and we're going to use that as a kind of a hook to grab these out of the cell with some technologies. So things are a little more complicated than what I just showed. So actually the DNA that gets transcribed is much longer than that picture. So DNA gets transcribed into RNA, and that DNA that eventually produces a protein includes introns as well. So in this picture you see a gene with 5 exons shown in different colored rectangles. In between those exons are sequences that we call introns. Those are actually chopped out or spliced out and discarded by the cell, recycled by the cell. And the remaining parts, the exons, are the part that get translated into a protein. So that coding sequence on the previous slide was just the exons concatenated together. But an important feature of this, one thing that allows the cell to do this structure, is that Well, while you're in the process of splicing out and removing the introns, you can do different things with putting the exons together. You can combine them in different combinations. So that's called alternative splicing, and this is a very common phenomenon. When it was first discovered, it was considered to be a very unusual and probably rare phenomenon, but now we know that over 90% of human genes undergo some form of alternative splicing, meaning that even when you know the complete sequence of the DNA, and even when you know what gets transcribed into RNA, you still have more work to do to figure out exactly what proteins might be formed. So by forming different combinations of exons into different mature messenger RNAs, you can make different proteins from the same original gene part of the genome. So proteins themselves have structure, and these have been studied for many decades. Proteins themselves are the string of amino acids. Remember, these are long proteins comprised of amino acids, so long molecules comprised of amino acids, not nearly as long as DNA. Typical proteins are a few hundred to a few thousand amino acids long, and they form very complicated structures, but they have some secondary structure as well. So the two common secondary structures that proteins form are called beta sheets and alpha helices. So the amino acids themselves can form these sort of flat structures called sheets or beta sheets, and they can also coil up into these helical structures that are called alpha helices. And you see examples of those over on the left. And on the right, you see examples of many different proteins, actual protein structures, and there are thousands and thousands of proteins whose structures have been solved, that is, biophysicists have figured out what the mature structure of those proteins is, and their structure confers upon them their function. So this is why we study their structure. So it's important in understanding protein function to know which parts of the protein are, say, buried on the inside of that structure, and which ones are on the outside, which are probably the more active parts of the protein. So that's protein structure. So another aspect of molecular structure sort of aspect of structure is transcription factors. So even when we know what the genes are, remember every cell in your body has the same genes, but cells behave differently. Skin cells behave very differently from blood cells. So why is that? That's because different genes are turned on, different genes are active in different cells. So one way that you control the activity of cells is there are proteins that are also, of course, encoded by your genome that go back and bind to the DNA itself, bind to those chromosomes, and can control the expression of proteins. That is, you can have a gene whose product is a protein, we would call this a transcription factor, that goes and binds upstream or downstream, usually upstream of another gene, and can accelerate the production of that gene or decelerate or inhibit the production of that gene. So these are called transcription factors, and they're a very, very important control mechanism that the cell uses to decide how much of a protein to produce. And finally, one other kind of important structure or mechanism is epigenetic structures, or epigenetic mechanisms. So epigenetic means sort of beyond genetics. That is, these are factors that are outside the DNA itself, the directly inherited material, that nonetheless control something about how your cell functions and how your genes are expressed. And one important, very important epigenetic structure is a methyl group. So DNA can get tagged with these methyl groups, then we say the DNA is methylated. And an interesting factor of this methylation is that it can get passed on through the cell cycle. So once you've tagged the DNA with this methylation, you can then, when the cell divides, the same positions can get methylated in the daughter cells that are produced from that. And these methylation marks, we now know, also are a controlling factor that affects how much of a gene or when a gene is expressed. So methylation marks are an important factor, and they are outside of the normal genetic factors in that they're not part of the DNA, but they are inherited, if you want to think of it that way, from one cell to another. And it's important to realize they're not inherited when a new individual is produced. They're just inherited as cells divide to produce new cells of the same type. So epigenetic mechanisms are another type of structure that we study and we talk about and use as a way of understanding the function of the cell.